Transcription is the synthesis of RNA from a DNA template strand. Since prokaryotes do not have a nucleus, transcription occurs in the cytoplasm and allows ribosomes to access the RNA strand as soon as it is created and begin translating the code into proteins. The process begins with a strand of DNA with a gene that needs to be transcribed. Before the gene, there is a promoter region, which includes the -35 region and the -10 Tata box. The -35 region is where the sigma factors bind to the DNA and calls for the polymerase core to attach and begin transcribing. The core and the sigma factor make up the holoenzyme, which is needed as a whole unit to initiate transcription at the site. At the Tata box, the RNA polymerase begins to unzip the DNA to form the transcription bubble so that transcription can occur at the +1 start site. Without the Tata box, the polymerase would not be properly bound and would not unzip the DNA strand and synthesize the corresponding RNA strand for translation. This would cause a lack or complete absence of the enzyme needed, and for example, in breaking down and metabolizing sugar, without the enzyme, the body would not be able to metabolize the sugar and would need to rely on a different energy source than glucose. For initiation, the sigma factor is no longer needed, so the polymerase core continues in elongation without it, and synthesizes an RNA strand towards the right based on the template DNA, not the coding DNA strand. The coding DNA strand is almost identical to the transcripted RNA strand, except it includes thymines instead of uracils. The polymerase adds ribonucleoside triphosphate to the 3' end of the RNA strand and cleaves 2 phosphate groups from the incoming RNTPs to use for energy. When the gene being transcribed ends, the polymerase dissociates from the DNA strand by either row-independent or row-dependent termination. Row-independent usually occurs if there are inverted repeats present on the DNA sequence near termination. The inverted repeats cause the RNA strand to fold on itself into a termination hairpin and causes a pause in transcription. The hairpin is followed by a string of adenines on the DNA template strand, which has a weak bond with uracil and allows the RNA polymerase to release from the DNA strand. In row-dependent termination, a row attaches to the newly synthesized RNA strand and races to the 3' end inside of the transcription bubble. This pulls the RNA transcript and DNA template strands apart, releasing the polymerase and ending transcription. This is like reading a book, because there are people that promote the book like friends and teachers at school, and this is where your mind latches onto the idea of reading the book like the polymerase. You go to the library to get the book and open the book up. This is the Tata box. You initiate reading at the transcription start site, and your eyes and brain act as the RNA polymerase. They follow the letters on the page to form words and sentences like synthesizing RNA and elongation to understand the story. When you get to the end of the book, you complete the story and close the book, and your eyes are done reading like the polymerase dissociates from the DNA template and RNA strands.